Yeah, so that's actually a very a very interesting experiment. So that was when I first made this microscope. It just starts to work, so I have very low expectations. I basically put the animal onto the stage, but I didn't see anything moving. But that was just because cells were moving at very slow speed. So I kind of just put the animals on and then went to lunch. And then when I came back later, I saw cells were moving crazy. So the picture, the video that's showing in the website is showing that it's near a tumor site, and there are leukocytes, which are the immune cells in your body. It's living red. So they are trying to combat the evil things the cancer cells are doing. So it's the immune response to the cancer. So what we captured there is that there is a tumor site, and then there is the entire vasculature that's running through the tumor, and the immune cells get the signals from the tumor cells, and then they came to rescue, either to support the tumor cells or to kill tumor cells, which is still a mystery. Either way, so they go off the vasculature, and then they start to form these clusters and do their own thing to combat or to support the tumor. So that's what the video was showing. I think it's a very powerful example of how advanced optical microscopy can reveal things that we didn't know before. And another amazing thing about that video is nothing was labeled. So it was just a rat with injected tumors, and all the signals we see, all the colors we see from that video is all intrinsic signals from the biological tissue. So what we were doing was merely manipulating the light to get the intrinsic contrast from the tissue. So I think that's really laying the path to clinical translation, because it's very hard to imagine you can do genetic manipulation to human beings to express fluorescent proteins, or even with injecting dyes, it's very hard to pass the FDA. So label-free imaging is definitely the way to go. It's just with that technology, it's very hard to get deeper than 1 millimeter. So we just need to attack it from another angle. How can we get these cool images, but deep inside the living animals or living patients? When we were doing this imaging, so this was my PhD study with Steve Bopard in UIUC. So when we're doing this imaging, the more we do this imaging, the more scared we are. Just how advanced, how smart these cancer cells are. They send these soldiers. We also visualized another thing that was not usually visualized. They are called extracellular vesicles. So these are just really, really tiny soldiers that can pass all these barriers with no problem. They can just go anywhere, do their dirty laundry for cancer cells at their will. So there are a lot of things happening in the cancer environment, and a lot of them are dynamic. So visualizing these things in living bodies in the intact context, I think is very important for both therapy strategies and also understanding the mechanism.